Welcome to Creativ's presentation. I'm Ron Baker. Creative is seeking Series A funding for $15 to $20 million to commercialize our revolutionary LifeTrack DX liquid biopsy for cancer testing. Even though cancer is the second leading cause of death in the United States, there's a very limited number of non-invasive methods to screen for cancer and monitor treatment response. Surrounding the tumor cells at the tumor site, there are many stromal cells, such as macrophages, T cells, and B cells. Scientists are well aware that tumor cells and tumor DNA enter the bloodstream from the tumor site, but have seemingly ignored the stromal cells that also enter the bloodstream. One type of stromal cell we observed and named cancer-associated macrophage-like cells, or CAMLs for short, we discovered provide very useful clinical information. These CAMLs are macrophages which have engulfed tumor cells at the tumor site. Our solution to patient management is focused on CAML cells in the blood. CAMLs can provide the information about the tumor. In addition, they can provide information about a patient's response to therapy. CAMLs are very funny-looking cells you probably have not seen in textbooks. The white arrow points to a white blood cell. As you can see, CAMLs are very large compared to white blood cells. They range from 25 to 300 microns in size. And they are polynucleated, and the larger the CAML, the more nuclear material it contains. Because they engulf tumor cells at the tumor site, they contain tumor proteins. Therefore, they are applicable for companion diagnostics for cancer drugs, such as PD-L1 is for immunotherapies. CAMLs contain tumor DNA, which is much more useful than circulating tumor DNA. CAMLs are also found in the blood of all solid tumors that we've analyzed. And CAMLs are found in all stages of disease. However, they are not found in the blood of healthy individuals. We capture CAMLs and CTCs using cell-sieve microfilters that we also manufacture. The scale for the filter and the cells are the same. The filtration process rapidly eliminates all red blood cells and the majority of the white blood cells. The filtration process is also very gentle, so as not to damage CTCs and CAMLs. We have performed a broad spectrum of clinical validations. This is a list of the 20 different solid tumors in which we have found CAMLs in the blood. In addition, we have participated in over 20 different clinical trials with pharma, biotechs, and academic institutions. We are funded by 3 research grants for cancer screening: breast, lung, and prostate. And we have 17 peer-reviewed publications, and CAML utility has been independently validated by 5 other groups, most recently 6 months ago by the University of Missouri. The clinical applications are very broad, as listed here. CAML size provides prognosis. If a single CAML larger than 50 microns is found in 7.5 mL of blood, the patient has a much shorter progression-free survival than patients who only have small CAMLs. We have developed companion diagnostics for more than 20 drug targets. We've also shown that CAML properties can provide information about treatment response within 2 to 3 months of the start of therapy. We are funded for cancer screening for single cancers, but we also have data showing potential for pan-cancer screening. And many copies of tumor DNA in CAMLs are available for sequencing. On this slide, the light blue indicates the applications of LifeTrack DX test from screening to cancer recurrence. For this round of funding, We will focus in the areas circled in red. The details are presented in the next few slides. When cancer is in remission, there should be no camels. We will implement LifeTrack DX to detect cancer recurrence in patient samples in our CLIA lab. If camels are present, the size of the camel can also provide information about the aggressiveness of the tumor. We will apply for FDA approval for companion diagnostics for immunotherapy for non-small cell lung cancer. Currently, all the FDA-approved companion diagnostics for PD-L1 require tissue. Tissue biopsy for lung cancer is difficult, expensive, and risky. In fact, our CEO's brother-in-law died during a lung biopsy procedure at Mass General Hospital. For the same FDA clinical trial, we also seek approval to predict treatment response. Imaging is used to determine disease progression. Immunotherapy, however, often presents pseudoprogression, where the tumor grows bigger when the patient is actually responding, because CD8 plus T cells enter the stroma. Camels can provide more accurate, objective information. Once we obtain FDA approval for our initial indication in non-small cell lung cancer, we will then target pan-cancer screening. This market represents a huge opportunity for us and will require additional capital to fund the costly clinical trials which are necessary for approval and commercialization. This slide provides a comparison of Camel's unique broad capabilities and advantages over liquid biopsy markers such as CTCs, ctDNA and methylation, and traditional protein biomarkers. Some specific examples include CTCs are found in small cell lung cancer in late stages of breast, prostate, and colorectal cancers, but are not usually detected in earlier stages of these cancers. Rarely are CTCs found in any stages of other cancers. Methylation signature, published data in Cell-Free DNA in 2020, demonstrated pan-cancer screening for 50 cancers. However, their ability to detect major cancers, such as breast, prostate, and kidney, is less than 10% in stage 1. And finally, ctDNA can provide companion diagnostics only for mutations and not for protein markers, such as PD-L1 for immunotherapy. The next few slides will address the market potential. The use of proceeds will include marketing activities and operational support for the growth of our CLIA lab, automation of the blood sample processing and artificial intelligence for reading slides, initial FDA clinical trial for companion diagnostics and prediction of immunotherapy treatment response for non-small cell lung cancer. The market size for cancer recurrence alone is very large. Currently, there are over 10 million cancer patients in remission. These patients typically undergo X-ray imaging 2 to 3 times per year. Achieving 1% market penetration alone will be over $200 million, and we expect to exceed that by year 4. The reason for applying for FDA approval related to treatment is for the LifeTrack DX to be included in the National Comprehensive Cancer Network's guidelines for standard of care. Physicians follow NCCN guidelines. We project that LifeTrack DX will be adopted in over 10% of non-small cell lung cancer therapies within 2 years after the FDA approval. The revenue for this segment is estimated to be at least $200 million. As shown here, the revenue projection is in hundreds of millions of dollars per year. We expect to break even before year 3 and continue to grow rapidly as we develop other products. We believe that we will become a multi-billion-dollar company. On our leadership team, Xiaomei Tang is the founder and CEO of Creatv. Pete Amstutz is the CFO. Incidentally, both got their degrees, all of their degrees, from MIT. I'm Ronald Baker, and I lead our business and strategy development with broad experience in cancer diagnostic products and services. Daniel Adams is the director of our clinical R&D. He discovered the clinical applications of camels and leads all of our clinical research. We wish to thank all of our collaborators, advisors, and customers, who without their close collaboration, we would not have been able to collect this valuable clinical data. In summary, LifeTrack DX liquid biopsy represents a revolutionary change in cancer patient management, and we look forward to presenting to you our exciting clinical data in detail. Our contact information is shown here, and our website is creativebio.com. Thank you very much.